This is Immuno Africa, a podcast dedicated to spotlighting African immunology research.
Did Canada need immunologists or Africa need immunologists? And the answer was clear for me. We need immunologists in Africa. In Canada, we have too much immunologists. I have too many colleagues in immunology in Canada. So that is what decide what I'm doing here now. My place is here. People need my work here.
Welcome to the Immunology in Africa podcast. I am your host, Welt Okete. If it's your first time here, kindly support our work by subscribing to this podcast and turning on notifications to keep up with new episodes. We're currently on Spotify, Apple Podcasts, Google Podcasts, YouTube, and other streaming platforms. In this episode, Dr. Gatien Lokossou talks about his decision to return to his home country, Benin, after training in France and Canada. Most of his work, which has focused largely on pregnant women and infants, explore preeclampsia, a pregnancy-associated disease that can be fatal, and breast milk. By way of introduction, Dr. Lokossou provides an overview of his educational background and how it led him to exploring the immune system.
Thank you so much, my dear Wef. And let me also thank you again for what you are doing for immunologists, but for all African scientists. So thank you so much. I landed in France in 2001 for my study to begin university. And at 2005, I get my bachelor. Then after that, I began my master study in immunology and vaccinology. And to get my master, I need to do an internship. And for this internship, I work on how epithelial cells respond to bacteria like E. coli. And then I began my PhD study on malaria, focused on how our genes are involved in our immune response, especially in pregnant women and newborn. I did it in France and in Benin. We were involved in recruiting pregnant women, almost maybe 600 pregnant women, and we collect the blood and measure antibody. We make also genotyping to find some single nucleotide polymorphism, but also haplotype, which can shape the immune response against malaria in newborn, in children, but also in pregnant women. And after that, I went to Canada in Montreal for my postdoc. After that, I decided to come back to Benin, and I get a position at university as lecturer and researcher. Currently, I am the head of the Department of Human Biology Engineering at University of Abomey-Calavi.
Okay, so thank you so much. So I want to quickly confirm. So all of your training, your BSc, MSc, and then your PhD, you took them in France, right?
In France, yes. You are right. Yes.
Interesting. But now you are in Benin Republic. So how was it like?
I'm just your neighbor.
Yes, yes, that's true. So how was it like transitioning from your training in France, your postdoc in Canada, and then coming back to your home country to do research?
When I was finished my postdoc, I asked myself, did Canada need immunologists or Africa need immunologists? And the answer was clear for me. We need immunologists in Africa. In Canada, we have too much immunologists. I have too many colleagues too many colleagues in immunology in Canada. So that is what decide what I'm doing here now. So it is clear for me in 2015 that my place is here. People, students, people need my work here, less than Canada. So that is why I'm here now.
I think I just like the fact that you mentioned that you had to relocate to meet a need. So you realized that, okay, Africa needs immunologists more. Africa has less immunology, so we need immunologists more than Canada does. And so you felt it was important to come back to your home and, you know, support research, support immunology, and tackle challenges in your country or in the continent, which is really, really beautiful and inspiring. So
Thank you so much.
yeah, you're welcome. So
but it's not easy, but we are going ahead.
Yes, yeah, I understand that part. Yeah. So so your initial training was in biology. So what got you, or what triggered your interest in the field of immunology? I know you said your master's was in immunology and vaccinology. So I want to believe there was
Yes.
like a kind of point when you had a point where you had to make a decision about what am I going to do for my master's, and then you decided to study immunology and vaccinology. So what triggered that interest in the field of immunology for you?
Very nice question. And I told it last week to my students at university why I decided to become immunologist. I told them after my BS, I asked myself, what can I do now? Parasitology, bacteriology, or what? And then I realized that immunology is involved in parasitology, in bacteriology, in fungal study, even non-infection disease. So immunology is everywhere. When you are sick, you need to focus on your immune response. When you are healthy, you also need to focus on your immune response. That's lead my decision to do my master in immunology and vaccinology.
Interesting. So thank you for sharing. I think the next question I want to ask is, what has your research been about generally? What's like a high level overview of your research? What kind of studies have you been looking into since your master's until now? So maybe you can just highlight a few of them for us.
Yes, of course. Thank you so much for your question. During my master internship, my research was focused on how epithelial cells respond to bacteria like E. coli. You know, the first capacity, our first capacity to fight bacteria or parasites is on the mucosal area, but also it's especially epithelial cells. So I was interested to understand how these epithelial cells respond to bacteria. And at that time, it was E. coli. And after that, I decided to work on one of most important disease in Africa, malaria. So as I would like to become immunologist, I decided to understand how the immune response was shaped by our gene. And this was very interesting. We have published 3 or 4 papers about that. And we found that some cytokine gene with specific polymorphism leads to increase the immune response, like antibody production against specific Plasmodium falciparum antigen. You can see during my PhD, my work was focused on pregnant women, and I get special interesting to work on pregnant women. So this lead me during my postdoc to work on a pathology which affects pregnant women. You can guess if it's preeclampsia, one of important um, pregnancy associated disease, pregnancy. And this could lead to eclampsia, and the result will be the death of the pregnant woman, but also for the fetus and the newborn. So my supervisor allow me to work on this disease, and our objective is to understand how the baby who have a part of his father between his gene could be allowed in the mother wombs. You know, the baby gene is a part of his mother and his father gene. And then we would like to understand how the human response is suppressed to allow the newborn, but also to be ready to fight pathogen. This is the paradigm of the pregnant of pregnancy, because when woman get pregnant, she need to protect his fetus, his baby, but also she need to be able to fight pathogen. And I was focused on how the immune response is modulated to allow the newborn, the fetus, but also to allow the mother to respond to pathogen. And we found that specific molecule such as called syncytin are expressed on placenta and are also found on specific microvesicle called exosome. And this molecule, syncytin, we have two kinds of this molecule, syncytin 1 and syncytin 2. Syncytin 2 are more important, are more involved. Our results show that are more involved in this immunosuppression during pregnancy. And then we decide to understand how this molecule affects immune cells, such as T cells, and how syncytin 2 reduce inflammation during pregnancy. Because the difference between normal pregnancy and preeclampsia pregnancy is the increase of inflammation, important level of inflammation. And our data showed that during pregnancy, we have high expression, normal pregnancy, we have high expression of syncytin 2, which leads to reduce inflammation against fetus. But in women with preeclampsia, the level of syncytin is reduced. Therefore, the inflammation is not well controlled, and this inflammation leads to stillbirth, lead to too many complications, such as death of both of women, but also the newborn. In normal pregnancy, both of these molecules are expressed.
Okay.
But syncytin 2 are more involved in immune suppression than syncytin 1 and other molecules. But during preeclampsia, the level of this molecule are less expressed. The level is reduced. So you will have more inflammation because you have less expression of syncytin, especially syncytin 2. So for normal pregnancy, we need to have important expression of syncytin 1 and syncytin 2. We need more expression. And what is interesting is that these molecules are embedded in microvesicle, such as exosome.
Yeah.
And this vesicle affects immune cells and reduce the capacity to produce cytokine, pro-inflammatory cytokine. So we after that, we are able to collect this exosome from blood, and we can measure in this exosome the level of expression of syncytin 2. And then we can know that these pregnant women are at risk to develop preeclampsia or not. This leads us to do a study here in Benin at 2015 to 2017, and we have recruited a lot of pregnant women at the beginning of pregnancy, maybe the first or the 2 weeks of pregnancy, and we collect blood from them until 20 weeks, because from 20 weeks you can find the particularity associated to preeclampsia if the women are at risk to develop preeclampsia. So we have collected blood from the beginning of pregnancy until 20 weeks of gestation. And then from this blood, we purify exosome and measure syncytin 2 level in this exosome to try to find which women are at risk to develop preeclampsia during the pregnancy. This is very important because it's like a kit to predict the risk to develop preeclampsia during pregnancy.
So basically, I'm saying both syncytin 1 and 2 are involved in suppressing inflammation in pregnancy. Yeah.
Yes, immune response, especially inflammation.
Yes, but it's syncytin 2 that plays kind of a more important role and is implicated in preeclampsia. So what your study did was to basically find out or help us identify women that would be at risk of developing preeclampsia by measuring the levels of these molecules by
in blood.
Yes, in blood via the exosomes and the vesicles that carry them. So I think it's really an interesting study with immense impact, right? I think I just got that big picture now view of the study being targeted at helping us to identify women that will be at risk of developing preeclampsia, in a way trying to spot at-risk mothers and fetuses beforehand. So if there were any ways to prevent or reduce the risk of that development, they could be explored.
Yes. If you have tools to early identify person who are at risk to develop a disease, you will do what you have to prevent.
Yes.
So our idea at that time is to develop a kit to measure easily this molecule called syncytin 2 in pregnant women. So when you find that this woman at risk to develop preeclampsia, but you will focus, you will take care about this woman during the pregnancy. You will do specific management, because when you have the risk, so you can use some drug to reduce inflammation. You can use some drug to avoid the complication associated to preeclampsia. Also, what we have found during this study, during preeclampsia, when you compare the placenta from normal pregnancy and the placenta from preeclampsia woman, the lattice is very small. The placenta from preeclampsia woman is smaller than normal pregnancy. Why? During pregnancy, one of important parts of the pregnancy is placenta. Placenta is involved in the in sharing molecule, food, nutrient from mother, between mother and the fetus, but also human cells, antibody, lots of things will pass from the mother to the fetus. Then, during preeclampsia, the placenta is very small, and this reduces exchange between mother and fetus. And we have also identified that syncytin are also involved in the development of the placenta, because these molecules are able to induce cells fusion. And when cells fuse well, the surface of placenta increase. So when you have less level of syncytin 2, you have less immunosuppression, but you have also less placenta development.
Thank you very much. Yeah, so you've worked on preeclampsia during your postdoc, but I think you are still in the context of working with pregnant women, working with pregnancy, working with infants. You've also explored breast milk, right?
Yes.
Great.
That is an important part of my research now.
Yeah.
You cannot imagine what you have in breast milk. It is it is a very, very nice liquid we need to explore.
Interesting. So I would like us to just pivot to that aspect of your work next. So I think what I love about the connection is that you are still working with women. You're still working with women who are pregnant or women who just got their babies, or infants. So let's talk about your research involving breast milk, and perhaps just tell us first of all what made you pivot to that work. What made you take interest in now exploring breast milk from preeclampsia, and what have you been doing so far working with breast milk?
Thank you so much, my dear. Thank you so much. I think you get the what lead my research: pregnant women and their infants. So when the woman get birth, how these infants grew up, what molecule, what particular thing are involved? That is my question in 2017. And then I decided to begin to work on breast milk, because I have the discussion with people, with other colleagues, and I have also read some publication. And then I have organized my research to collect breast milk. This is wonderful, free. It is free. You don't need to buy. This liquid is free. So in this biological liquid, you have things you can never imagine. You have human cells, you have non-immune cells, you have bioactive components such as cytokine, such as chemokine, lipid, hormone, too much thing inside breast milk. But you have also microbiota. You have particular bacteria, particular virus in breast milk. And all of these are involved in the shape of the immune system of a newborn. This is very wonderful. That is what led me to work now on breast milk. And currently we are characterizing bacteria in breast milk, but also I will tell you later how breast milk could protect newborn children from disease. One of the study we did was conducted by one of my master's students, and this student has showed that we have in breast milk particular special bacteria which can fight pathogen bacteria. When you have pathogen bacteria like salmonella or Escherichia coli, the composition of breast milk, particularly the composition of bacteria, help to fight this pathogen bacteria. Another student, bachelor student, find that, of course, we have bacteria, but in the serum from breast milk, we have other components. We have bioactive components, which we need to characterize now, which could destroy bacteria. What she did is very simple. We get breast milk, then she has centrifuge it and collect the supplement, which is like serum, and then she puts some of this liquid on pathogen bacteria, and 2 days, 3 days, or 4 days after, she found that the growth of bacteria reduce. So when you breastfeed your infant, you can protect him from bacteria infection like enterococcal bacteria and other. And now we are designing a study to understand how breast milk could be used to protect infants from malaria, because two study have showed that in breast milk they found that there is antibody against some bacteria antigens, and then we are designing a study to understand how human cells in breast milk could fight this antigen, how this cell can produce antibody in breast milk which can fight malaria, because now we have evidence that malaria antigen, Plasmodium falciparum antigen, are found in breast milk. So if this antigen is in breast milk, but the immune cells present in this milk will do something. Now our next study will try to explain how human cells found in breast milk fights Plasmodium falciparum involved in malaria.
I feel like the first thing that comes to mind is it possible that breast milk could have all the negative, have some negative impact on infants? So, for instance, can breast milk spread disease between a mother and her infant, or maybe are there any other concerns, negative concerns associated with breast milk? So that's my first question.
Oh, okay, so let me answer. But before, let me tell you that a breastfeed infant have has a reduced risk of infection disease, but also non-communicable disease such as asthma, cancer, autoimmune disease, obesity, and other.
Yeah.
Okay, so now imagine a woman with HIV. Some study have shown that you can find HIV particle in breast milk. So it's not good to breastfeed your infant in this case. Also, when you have inflammation of mammal gland, this could affect your newborn. When you have some bacteria of virus infection in the mammal glands, you could not feed your infant. But
Yeah.
if you are not sick, if you are well, you don't have disease, it's a mistake to not feed your children.
Thank you so much. So you've highlighted HIV and the possibility of other viral or bacterial infections related to the mammary gland that could, cause pathogenic organisms to be in breast milk. So in those cases, it will be unsafe and unadvisable to feed your infant breast milk.
Let me add another thing before.
Okay.
When the mother, because I told you later that if you have mammal gland infection, you are not. It's unsafe to breastfeed your children. But if you have other disease, infection disease, okay, and your immune response allow you to produce antibody or immune cells to fight this disease. This antibody come to the breast milk, and then you can breastfeed your children to protect him from this infectious disease, such as, I don't know, malaria or gastrointestinal infection, something like this.
Okay.
So I don't know if you get it. It is different from disease associated with the breast directly.
In the context of someone who might have HIV or someone who might have some of these infection of the mammary gland, and perhaps they want to explore formula food for their infants, I'm just wondering what are like some of the differences or similarities that could exist between formula food and breast milk, and then whether how closely related are the two options for infants and for women?
In formula food, you do not have human cells. You do not have antibodies. You do not have cytokine. What you have only molecule to grow up, but nothing to shape his immune system. In breast milk, you have human cells, you have cytokine, you have antibodies, but you have also nutrients to help your newborn to grow up. The choice is clear and easy. We need to promote more the breastfeeding of newborn and infants. We are far from to understand all molecule, to identify all molecule in breast milk. And yes, and today I'm sure that we are not using all benefit from study on breast milk. All data available, we do not benefit from them. We have this data, but public do not know, do not use, do not understand what we are doing. So we need to make a important promotion of. We need to share the data in. Not only in scientific publication, but also non-scientific publication, radio as what you are doing, but also newspaper, and talk about breast milk because it is free. You do not to pay for that. It's a gift from God.
That's very true, and you're very right about the need for more public engagement, community engagement, science communication, mass media communication to just amplify the immense benefits that breast milk offers. So we are making use of maximizing its benefits, maximizing the great blessing that breast milk is to us as humans. So I'm just wondering, because you said the breast milk has all of these benefits, immune cells and lots of benefits, and then of course you said it's free. But how does the mother's nutritional status, the mother's immune status impact on the breast milk composition? So for instance, someone who is immunocompromised, or someone who doesn't feed appropriately, how will that affect the breast milk composition and by extension the infant? Of course, particularly in the context of these immune components, the immune cells, the cytokines, and some other components. So how will that affect? Because even though the breast milk is free, I'm just thinking that maybe we also have a responsibility that we are able to preserve or maintain the composition of this breast milk.
Great question. Thank you so much for this question. You need to have an appropriate nutrition to allow a good composition of your breast milk. So, of course, if you take some drugs, you cannot breastfeed your children. So, first, we know that when you're a pregnant woman, do not appropriately eat or do not have appropriate nutrients during pregnancy, this affects the immune development of a newborn. So imagine this woman, this pregnant woman, she gave birth, and then after she don't have enough nutrients, she don't have appropriate nutrition. She will affect the human development of her newborn. So we need to pay attention on nutrition, but also on all conditions which can affect a good breastfeeding.
Thank you so much for that contribution. So the fact that mothers also have to pay attention to their health status, the strength of their immune system, and then their nutrition to ensure that this composition of the breast milk is preserved, and then that infants that they feed can get the maximum benefit from consuming breast milk, right? Yeah.
Yes.
Yeah. So thank you so much. So I'm just wondering if there's any other thing you wanted to chip in about your work before we take maybe the last few questions.
In our continent, we have a great problem with research funding. We need to explain to our government what is the importance of research, not only on breast milk but all research. Guess what? I don't know if it's the same thing in your country, but here in Benin, students need to pay by themselves all reagents they need to do the research if the project is not granted. And you know the percentage of projects which are granted in Africa, and it is not enough. So our government need to fund research. And then we can contribute to make people healthy, and so and so and so.
Thank you. Thank you for that contribution. So I love the fact that it was straight to the point that the government has to fund research and, help our scientists to even the younger scientists in school to, make some of these discoveries that will benefit society, that will benefit our people and, contribute to tackling diseases and improving the health status of Africans and of our own people. I want to ask this really interesting question about what has been the most important decision that has shaped your career or shaped your life, or maybe not a decision, but maybe an advice that you received from someone that has shaped your life or your career.
Yes, thank you so much, Biff. It was an advice from a good friend who currently is in Canada. We were all in France before, and then he went to Canada for working. And when I have finished my PhD, he told me, "Come to Canada. You will see. You will have a lot of opportunity to do your research here." And I replied, "Are you sure?" He said, "Yes." And this allowed me to do my postdoc, but also to have another view on research. And I think his advice was very important. It's not a scientific advice, but it's a friend advice.
Great. So we're wrapping up the conversation already. And I think something that strikes me about your work is the consistency that you are focused on women, fetuses, and infants. And yeah, that's really interesting. So well done.
Thank you so much. Thank you so much, my dear.
So now I think I wanted you to share like a closing remark about yourself, about your work. What would you want to be the take home for listeners on this episode?
Especially for students, never give up. Never give up. Do all is close to you, all is depend to you for your research. And then we need to talk with our government, with all political decisions. We need to be involved to improve our capacity, to improve our student capacity in research. They have a lot of capacities, but we are not using it now because we don't have money. They don't want to. They want to do a lot of things. When they come to your lecture, they discover explain them in immunology, how cell growth, how immune cells are shaped, and after that they come to you. Professor, I want to do this with you. Is this possible? Yes, but now we don't have funds. We don't have money. We need to ask for money. This is a great problem. This is a good problem. We need to solve that. We could not be independent nation if we are not able to decide what we want to do in research, what kind of research we need to fund. Yeah. We need to decide, and we need it. We need it. See what we are doing on breastmilk. At this session, 3 of my students a bachelor student work on breastmilk because I told about them during my lecture, and they are very interested. They came back to me and they decide to work on it. But we need money, we need reagent, we need too much thing, and we don't have. But we won't never give up. We will continue to work, and we will continue collaboration. We need collaboration. We have some collaboration in Germany, in Canada, and in France also, but we need to develop our collaboration.
I just to comment that I think you really shared important points, starting from the need for students, and I think by extension researchers to really immerse in their work and to make sure they are giving their best and putting in their best. but also calling to our governments and all the relevant agencies to fund research so that we can do the kind of work that will benefit our own continent and that will benefit our people, that our researchers will be able to answer the kind of questions that they want to answer and not the type that funders want them to answer or explore.
Thank you for listening to this episode on Immuno Africa. If you enjoyed it, please feel free to share it with your network. You will learn first about future episodes and get other immunology related updates by following Immuno Africa _ other Immunology in Africa podcast on social media. See you on the next episode. Bye.